This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 532, recorded on January 25th, 2019. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. We have clouds in the sky. We had some of the worst weather I've ever seen in Michigan this week, with a terrible icy morning on Wednesday. It rained at 32 degrees Fahrenheit, and on top of the remains of snow that was there. It was awful. So now it's just cold.
We had that storm, except it was 44.
Yeah.
And it was like tropical. And then that night, it all blew away, and it was freezing again. So all the water froze. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. We had an ice storm, followed by a rainstorm, followed by not too bad.
That's weird.
Yeah.
And from Austin, Texas, Rich Condit.
Hey, how you doing?
All right.
We got 53 Fahrenheit, which is 12 C, and I would say mostly cloudy, occasionally a shot of sunshine. Not too bad.
A shot of sunshine.
Yeah, a shot of sunshine. It blasts through my window.
Please take our TWiV poll. 5 minutes, 12 questions, asm.org/TWiV poll. So find out more about you. Anything new happening with ASV, Kathy?
Yes, we have news because of the shutdown. For those people who are directly affected by the shutdown, and you know who you are, we are extending the deadline for abstract submission to February 15th. We made a special arrangement with FASEB to do that. And after February 15th, we're not going to be able to consider abstracts for oral presentation because of the timeline in putting workshops together with everything else. But we can still take abstracts for posters after that time. So
is the shutdown going to be over by February 15th?
Nobody knows.
No, it may not be necessarily, but after February 15th, we can't extend our deadline beyond that. Right. That's all.
That's
so
true.
I was asking a sort of a snarky rhetorical question.
Right. Yes. Right. So the details are on the ASV page, on the home page. And then, of course, if you're on the University of Minnesota page about the meeting where it says submit an abstract, deadline February 1st, if you click on that, it goes right through to the FASEB page. And at the very top of the FASEB information page, it tells about the extended deadline. There you have it.
Okay.
All right.
Yeah, everybody else still have February 1st as your deadline, which is a week from today, right?
Yeah.
We're recording on Friday. Yeah.
Next Friday. All right, we have to get ours in. All right, speaking of deadlines, March 1st is the abstract deadline for the European Congress of Virology, April 28th through May in Rotterdam. ECV2019.com is the URL for that. Okay, and the ASM Clinical Virology Symposium, May 5th through 8th in Savannah, Georgia. Abstract due May 8th.
March 8th.
March 8th.
That's what it says here.
March, you're absolutely
right. The conference is May 5th through 8th.
asm.org/CVS. Boy, there's all these abstract deadlines, right?
Yeah. Then
we have the Intel International Science and Engineering Fair. They need judges. They need PhD and MD Grand Award judges in 22 scientific and engineering disciplines, including biochemistry, cell and molecular sciences, biomedical, and health and microbiology. There will be a link in the show notes because there's no point in me saying it because you'll forget it.
Right. But our friend Trudy applied and was accepted, so she's excited. She's going to Phoenix to do this judging. Cool. Yeah.
Nice. All right, we did our job.
Mm-hmm.
And just a bit of news to follow up on something we talked about earlier, an article in Science from January 21st notes the scientist behind the CRISPR twins sharply criticized in government probe loses job.
Good.
Yeah.
Yeah, preliminary findings. Oh, you know what he did? He faked all the documents, the ethical review documents.
Yeah, some of the.
I didn't know that. Wow.
Because he wanted to be first. He wanted fame.
He's just off the rails.
Yeah.
This guy is crazy.
Yep. All right. So we have a little snippet from a listener.
We'll see if this turns out to be a little snippet.
Yeah. Someone wrote, really?
Yes, that was me.
Is that, yeah. Alan, you have adopted Red. Is that
Yes.
right?
Alan's Red.
Okay, good.
Tomas writes, hi, first of all, thank you for your iTunes U lectures and TWiV. They helped me a lot when I was learning microbiology years ago. In my country, Poland, there is a heated debate going on currently with both online and offline protests related to wild boar population control and African swine fever. Perhaps it would be an interesting subject for TWiV. The issue goes on more or less like this. Wild boar population is getting infected with ASF moving progressively east to west. This in turn threatens the pig production, which is around 50% of all meat in Poland and Europe. Of course, it's important that farms have to introduce biosafety measures to prevent the pigs from getting infected. But if the disease keeps spreading through the ecosystem and wild boar remains a consistent reservoir, this may not be sufficient to maintain small and eco pig farms. Because of that, the Polish government has announced a mass hunt of wild boar to drastically reduce its population in areas touched by ASF. The protests are happening now, with ecologists saying that mass hunt is pointless because it will cause the disease to spread even faster. Boar moving further distances, trying to avoid hunters. Empty ecosystem providing place for more infected boar from the east coming over. Also, if population control of the wild boar is indeed effective, such a sudden removal of this species from the ecosystem could potentially cause a lot of adverse effects. Question that seems to be on everyone's minds are, can we keep the wild boar population and keep pigs farms safe just using good prevention methods? Are we perhaps at a moment where we have to decide between one of the two species, either keep the pig production and severely limit the wild boar population or drastically lower the pig production only to farms that can keep the highest standards and keep the boar population roughly intact, accepting that it carries ASF? The debate on the subject is very heated and political right now, and it would be interesting to hear anything about ASF on TWiV. I would like to have that heated discussion right now in the U.S. We don't have anything so interesting. Right.
Yeah. The one thing we should clarify before we dig into this, the Eurasian boar, the wild boar that Tomasz is talking about, is a native species in Europe and Asia. Mm-hmm. That's why it's the Eurasian boar. It is an invasive species in the U.S. And in the U.S., we have feral pigs that are a mixture of Eurasian boar and the domesticated version of those, which is the domestic pig, and that the occasional escapes of domestic pigs crossbreed with the wild ones, and so there's just this general population of pigs wandering around in the wild that we refer to as feral pigs. I'm sure there's some of that going on in Europe as well, but at least in Europe, we're talking about a species that was there even before humans came along and started rearing domesticated version of them.
Mm-hmm. So how many boar do we have here? We have lots?
Millions. We have a massive infestation of feral pigs in the United States. It's, Rich may have encountered this. It's huge in Texas, but all through the Southeast up into the Carolinas, and they have started to move northward, possibly because the climate is warming. And also they're getting more acclimated to human sprawl type environments. But there is a huge problem with invasive pigs in the U.S.
And where did they come from? Do you know?
Oh, they've been here since European settlers came and brought their pigs.
I see.
So there were escapees from that, and some of those were closer to the wild relatives than they were to the native. And there's also a compounding problem here in the U.S. where areas that haven't had a large feral pig population have suddenly started seeing feral pigs popping up, and they've been getting to places that they couldn't have gotten to without human assistance. Because hunting these has become a very, very popular and lucrative business. They're hunted for sport throughout the Southeast, and people who are enthusiastic about hunting pigs will bring a few to their home state, maybe Virginia, maybe Maryland, maybe New York, and set them free so that they can have a wild population of their own to hunt. And of course, then you've got a feral pig population in that part of the country.
Yeah. So we don't have ASV here, but the potential is that
we don't have ASV yet.
ASF. I'm sorry.
Or AS,
we do have ASV.
ASFV. Yes, we have ASV. Sorry about that. But maybe we will.
Yeah,
and
sure.
One of the important points, which I'm sure we're going to get into with that, is it infects the wild Eurasian boars, but they mostly survive it. When it infects domesticated pigs, they mostly die.
Right. So this is a pox virus, and I thought that Rich Condit might start out by telling us a little bit about the virus.
Okay, I would say it is a poxy virus.
Poxy.
So
it's very sticky when you mix two parts of it together.
That's great.
It is among the nucleocytoplasmic large DNA viruses, and it's one of the more, at least historically, I would call traditional ones with a smaller genome. These things get really big. This is more poxy size, with 150 to 200 kilobase genome, double-stranded DNA genome, covalently closed ends, and replicates in the cell cytoplasm, same as pox viruses. So it carries all its transcription machinery in the virion and does everything in the cytoplasm. When you get down to it's a little different looking than pox viruses. It's icosahedral rather than brick-shaped, and it actually has retains a protein coat on the outside that gives it the icosahedral symmetry, and underneath that is a membrane, and underneath that is a core. Pox viruses, in their assembly, go through a phase where the membrane is assembled on this, what looks for all the world like a capsid scaffold, but then the capsid kind of goes away. And I think of the swine fever viruses as similar, but not losing the capsid. So they have a core and then a membrane and then a capsid. And apparently they can acquire a second membrane, much the same as pox viruses as well. So in many respects, the biology is quite poxy. But when you get down to looking at the similarities at the nucleotide level, there's considerable variation, and they don't cross-react with each other and that kind of stuff. So its closest cousin is the pox virus is in the NCLDVs, but it is its own thing.
Yeah, it's a different family. I'm sorry, it's not a pox virus. It's an Asfarviridae.
Yeah.
I like this new adjective, though, poxy.
Poxy's great.
So my understanding, I'm going to debate with you on the European boar here, Alan. My understanding is these arose in Eastern Africa and have been there forever. And their natural cycle involves oscillates between warthogs and soft ticks. Soft ticks are a vector, and they replicate in the vector, and they transmit it to warthogs. And in both of those animals, the ticks and the warthogs, it can establish a persistent infection that is, from what I understand, asymptomatic. However, when it spills over into into swine of other species other than warthogs, it can be quite lethal, and I think that includes the wild boar, and certainly in domestic pigs with a mortality of up to 90%. It was sort of a disseminated disease that affects a lot of different organs. Go ahead.
Yeah, so there are a few different pigs in the world.
Yeah.
So the African warthog is, I just had to look this up, Phacochirus africanus. And that is, it's got a very distinctive kind of shovel-shaped snout that comes out with these two very pronounced tusks at the end. And I think that is what you're talking about. I think it actually has 4 tusks. That's what you're talking about, which is the wild or original or whatever term you want to use host, I think, for African swine fever.
Yes. That's what I'm talking about. Yes.
Right. So then in Europe and Asia, there is Sus scrofa, which is the wild swine, the Eurasian wild pig. That is also a native species, but it's native to Europe, Asia, and just a little piece of North Africa.
Mm-hmm. Okay.
That's distinct from the African warthog, but I gather at some point it must have had some kind of exposure, either in ancient times or more recently, to African swine fever, and is apparently resistant to being killed by it.
Okay.
So that's a second species of pig. The domesticated pigs are, as I understand it, a derivative of the Eurasian Sus scrofa, which has been domesticated and bred by humans in captivity for eons, and is now, the pink, very little hair pig that you see at the state fair. The Eurasian boars that we're talking about in Poland and that are dominating the feral pig population in the U.S. are Sus scrofa, and they're darker brown, but still look very, very pig-like.
The other issue is that although it's described that in the original host, the warthogs, the transmission is through the soft tick. Transmission in these other swine species is primarily through either direct contact or contact with contaminated materials. Mm-hmm. Okay, mm-hmm. And contaminated materials is a big issue. Carcasses or things that have touched the animals, equipment that's used to deal with the carcasses, boots of people that have been trotting around.
Hay.
Hay.
This is what surprised me, because I was looking at the picture on the viral zone with the envelope that's sort of on the outside, and anything that's like that to me screams not stable in the environment. So the fact that it could spread through secretions and survive for long periods of time on clothes and shoes and hay was surprising.
Right. That's why in our little back and forth here on the cheat sheet that we use to do the podcast, I said, this is not your grandma's envelope.
Right.
It's more, remember, we've discussed the poxvirus envelope before about how so poxviruses have an envelope, and I envision this as much the same kind of envelope. Poxviruses, you can lyophilize them and rehydrate them, and they're fine.
Yeah.
And I remember I've talked before about how you can actually even strip the lipid out, and the protein that's protein network that's in the membrane remains intact in the absence of a reducing agent. And if you strip the lipid out, you lose the infectivity, but then you can add the lipid back and regain infectivity. So this is not a typical membrane.
Right.
And the virus is actually quite stable, and that's an issue for the transmission as well. Yeah.
Just a different kind of exception for the students in my virology class.
And just one more exception. And there's no vaccine.
Right.
People have been working on it forever. And I don't know what all the issues are. I've seen papers throughout time of various attempts to make subunit vaccines and attenuated by passage live vaccines, or deliberately attenuated by deletion or engineering of one sort or another. And apparently they've not been successful so far. That's a real.
Is anybody working on engineering the pigs to resist it?
That I don't know.
Because we've got a I would think we would have a good lead on that since the wild boars are relatively resistant.
You would think.
Yeah, you got to figure out why. It's not straightforward. Vaccine seems to me the way to go, but
yes,
people have been working on it for a long time.
Absolutely.
And
at least 10 years.
And I've read in one paper, I think an article by Linda Dixon, she wrote, we're not quite sure what the correlates of protection are. So that's a problem if you don't know what it is that you need to induce. But that would be the thing I would say. If you had a vaccine, you could drop it in bait in the forest, very much like we do for rabies.
Well, if you had a vaccine, you could just vaccinate the whole domestic herd.
I guess you could do that. But if you wanted to hit the boars, right?
Right.
You could vaccinate.
But if the domestic herd, you've got them.
Yeah,
and
you could do that.
And they're already vaccinating them against other stuff like classical swine fever. That would be the easy solution. And you could go after it in the wild, but that wouldn't really be as essential.
So there's no shortage of articles if you search the literature. I have a couple here I want to point out. One is it's got a .nl top level domain, which is Netherlands, but it's Agroberichten Buitenland. Anyway, it's the African swine fever in Poland does not let go. So this is interesting because 2 outbreaks in southeast Poland in small backyard farms, one with 1 pig and a second with 4 pigs. And there is bad because you have no biosecurity whatsoever, right?
Right.
And the farmer walking through the woods, maybe the boots get contaminated and walks in and contaminates the pigs. So that's very easy to happen. Now, Kathy picked up on something here that I will let her tell you because it's pretty funny.
Right. So I'm just quoting. The newly appointed Minister for Agriculture in Poland canceled the idea of building a fence along the eastern border of Poland. This is not going to solve the problem.
No.
Pigs will not bore. So this photo here in this article, there's what looks like a pig with black hair and a very pink nose. Is that a boar? Or is that a pig?
Is well, I think a boar is just a male pig, and I think that's just a regular domestic pig.
So not all boars have the tusks, right?
I guess not. I don't know what the... Right, so the African warthog, separate species, it has tusks. The wild boar, I think, develops some tusks in the male.
Oh, wait, I have something here from livescience.com, pig facts.
Okay.
A boar is an uncastrated male domestic pig, but it also means a wild pig of any gender.
Mm-hmm.
A hog often means a domestic pig that weighs more than 120 pounds. Pigs are also called swine.
Mm-hmm.
Okay, I think the males may grow their tusks if they're uncastrated.
Maybe.
Mm-hmm. Okay.
I either read that somewhere or saw
made it up.
Or made it up. That is always a possibility too. But but I think the tusks are not found. The other thing is domestic pigs are killed at some ridiculously short time frame nowadays, like 10 weeks or something.
Mm.
So they may just not get old enough to grow tusks.
So in this article, they talk about biosecurity that you would need, and a lot of big pig farms do that. They are very strict. Yeah. I remember once we were in Western Canada, and there were big pig farms there, and my daughter wanted to go see a pig, and they said, no, we can't let you in, and we can't bring them out because then we wouldn't be able to bring it back in again. They're really strict about that. And this, of course, affects pork production because if you get infected pigs, you can't sell them.
Right.
So in this article, it says that many countries have introduced a ban on the import of pigs and frozen pork from Poland. So that's a big problem. And then there was an article in Science magazine from 2017. African swine fever outbreak alarms wildlife biologists and veterinarians. And they have a photo here of pig farms somewhere culling their herds.
Wow.
To prevent the spread of the...
They're just stacking up dead pigs on a pile of tires, and I guess the tires are there to accelerate the blaze when they light this whole thing.
probably. Do the same thing when they have an outbreak of foot and mouth disease. They did that recently in the UK. They killed all the cows, basically.
It's a huge waste of well, not waste, but huge loss of protein.
Mm-hmm. Yeah. And of course, these boars cross borders, right? So it just keeps spreading. And it's a problem. Yes, Kathy.
I was just thinking that destroying the tires destroys the mosquito breeding capabilities.
The tires.
But I think this is happening in Russia, so maybe not an issue. This picture.
ASF first struck Europe in 1957 in Portugal from Angola. So this Spanish outbreak there was an outbreak in Spain. It took 35 years to end.
Yeah.
How did they end it? Does anyone know?
Let's see how
it was largely culling animals and controlling as best they could the wild pig population. But it's largely culling animals, I think.
By the way, in the science article, they talk about how Lithuania built fences and fortified their border in 2013 to prevent the virally infected pigs from spreading in. And then about a year later, they found that, in fact, the virus was in Lithuania.
Yeah.
Probably the pigs can get through the fences, right?
Oh, they dig.
They dig.
And
they go under.
These are borders that go through woods, and how sturdy a fence are you going to build? And they're going to get through.
22,000 pigs slaughtered in 2015 in Estonia. Virus in wild boar complicates it. Feces, urine, nasal secretions from the boar contaminates soil and plant material, which dog walkers or mushroom pickers might carry out of the forest. Great. Hunters, too. Blood is infectious. Family members seem to infect one another. And there's a map here showing some of the populations in Europe. So, they say the surest way to get rid of ASF and wild boar would be with a vaccine packaged in baits.
Okay.
So, Kathy, you had noticed something here, too.
Yeah. Well, let's see. Almond-flavored balls of fat and corn flour covered with soil. Why almond-flavored?
Yeah.
They like almonds.
Do you think they like almonds? Boars like almonds?
Yeah.
Sure. They're omnivores, but they do eat a lot of nuts in the wild.
Okay. All right.
I forget what they use in this sort of vaccination in the wild we've talked about before for controlling rabies.
Right.
I forget what they use as a bait there, but it's not almond flavored.
Well, for rabies, they use something that's kind of like a dog biscuit, I think.
Yeah, I think so. Yeah. Following up on how did the Spanish get rid of it? Just this one line from a review that Linda Dixon wrote. Following introduction of ASFV to ASFV-free countries, the only control measures available are strict quarantine and biosecurity, animal movement restrictions, and slaughtering of affected and exposed animals. Okay, so that's presumably a combination of those measures is what was applied to get rid of it in Spain.
Yeah, you wrote to Linda, and she wrote a nice. You want to summarize that for us? That would be great.
Just summarize.
You don't want to summarize. There's only 5. Maybe you could read it. There are only 5.
I can read it. I can read it real quick. So
what is Linda? What is Linda's position? Is she over?
Oh, right. Okay. Yes, she's a PhD scientist. An investigator working at Pirbright, which is Pirbright Institute, is a animal health research institute in the United Kingdom that works on a number of high consequence, high risk animal pathogens, including foot and mouth disease virus and African swine fever. And so she does a variety of both basic and applied studies. So her focus is African swine fever. Mm. So, I just sent her the email from our listener. Was it Tomas? Yeah, that we're talking about. And she makes 5 points. 1, ASFV has been spreading since its introduction in Georgia in 2007 through the Russian Federation, Eastern Europe, including 9 Eastern European countries, and most recently in 2018 introduced to Belgium and China, where the disease has spread widely since August. In the EU, spread in Poland and the Baltic states has largely been in the wild boar population. Some bigger jumps have occurred, for example, to wild boar in Belgium and the Czech Republic. In Poland, the spread has been mainly from east to west. The disease in wild boar is similar to domestic pigs. Okay?
Oh, so
and is fatal in almost all infected wild boar.
Oh.
That must be where I got that.
Okay, I was mistaken.
The carcass and around it remains as a source of infection for uninfected wild boar.
Oh.
So if you find a dead boar in the woods, just leave it.
Yeah.
Don't touch it.
And that also cans my idea of using wild boar as a model to find effective vaccines. So they're just as susceptible.
Apparently.
2, as ASF is a notifiable disease to the OIE, World Animal Health Organization, as well as national and regional authorities. So it is a notifiable disease. Notification of a new infected area results in quarantine, movement and trade restrictions in the infected area, and a surrounding surveillance zone. Man, they shut parks right down. Exports of pork or wild boar products and pigs from these zones are prohibited. So we're big red flags. In the EU, exports from outside these zones are allowed, but third-party countries may choose not to accept imports from an entire country. For example, I believe a number of agent countries do not accept imports from affected countries in the EU, even Belgium, which has never had a case in pigs, and has culled all pigs in the infected wild boar zone.
Bear in mind, anytime you talk about import restrictions, often something like this is used as a free excuse to protect your own local farmers.
Sure. 3, spillover of infection from wild boar to pig farms does occur, although smaller scale farms are more likely to be infected. Some large scale farms have been infected. The trade restrictions do not differentiate between the level of biosecurity practiced on farms and an introduction of ASFV into wild boar in a region will similarly prevent all farms in the zone from exporting products or moving pigs, i.e., pig farming cannot be carried out in an infected wild boar zone.
Wow.
4, the population of wild boar in many European countries is very high due to an absence of natural predators and favorable breeding conditions, amongst other factors. Corridors of wild boar movement have allowed ASFV transmission across boundaries and through countries. All countries carry out culling to try and control the population. Poland culls wild boar every year. An expert from the National Veterinary Research Institute was quoted recently saying the cull this year is not higher than previous years. The EFSA recently published a scientific opinion regarding African swine fever in boar populations. The opinion, she gives a link, the opinion concluded that in infected areas, shooting wild boar was not effective. That's important, as it encouraged them to move. However, reducing the population density ahead of the epidemic front could reduce the rate of spread. The epidemic causes death of many wild boar, and population is reduced in the aftermath of an epidemic, although ASFV is still present. The report also provides information about previous attempt to limit the wild boar population and concludes that sustained culling over the years is required to maintain or reduce the population. It is very unlikely that the cull in Poland will eliminate the wild boar. I have video from a friend in Warsaw showing a wild boar grazing on a green space outside her apartment. You may also find interesting a recent update on the situation in Belgium and impact this is having on many factors since a large area of forest is effectively shut. And she gives a link to that. I also note there's a conference in Germany next week on control of African swine fever by hunting. The Global Framework for Transboundary Animal Diseases, ASF Europe Group, has also published guidance on control of ASF in wild boar, including biosecurity for hunters. She gives a link to that. And I like this best. My final comments are, if you want a pig industry, you have to control ASF in wild boar. Currently, targeted culling seems to be the only option in the future. Currently, targeted culling seems to be the only option. In future, when we have a vaccination of wild boar using baits should help control the disease. So, big problem.
It is. It is.
And when it's my impression is that when this shows up, they shut down everything around. I mean, some of the other stuff that we posted that I read about, maybe it was this update that she posted. I mean, they closed parks. You can't go in.
Yeah. And in terms of hunting them by culling, it is correct that's effective if there's enough hunting pressure, but it's also probably never going to be possible to eradicate these. There are estimates, and I linked a page on the Virginia Department of Wildlife that has some good information about the nuances of this in the U.S. But apparently they modeled the wild boar population in the U.S. and found that you'd have to have a 70% killing of the feral hog population every year to continuously reduce the population size.
Yeah, I don't think that's happening.
And these animals are, they travel in groups called sounders, but they're smart enough that if a sounder gets cornered and a bunch of them get shot, they'll split up. And then you've got two smaller sounders of pigs that are reproducing on their own, and they're harder to track.
This Department of Game and Inland Fisheries thing that you posted from Virginia is great. Whoever wrote this hates feral hogs.
Yes.
the first sentence is, feral hogs are four-legged ecological disasters.
Yes.
Actually, they strongly discourage control by hunting. So it does not work. Yeah. Does not work. Actually leads to more feral hog. I guess because people decide it's a great sport. Let's get more hogs.
That's what I was referring to earlier. Now, Virginia's in a unique position because they're on the border of North Carolina, which has a very active feral hog hunting season and, not season, but community. And I think there are guides there that make money from guiding people to go shoot hogs. And there's Virginia immediately to the north that doesn't really have that going on and doesn't want it because, of course, these are very destructive. They destroy crops. They'll mow through a field and dig up everything. So Virginia really doesn't want this to happen, and they're discouraging hunting, but at the same time, they're trying to trap and eradicate as many of these as they can.
Well, and if all you need is an introduction of African swine fever into the country here, and we're in deep doo-doo.
It's likely to happen at some point. This is a very stable virus, so it's going to come over.
Right.
Yep, it's going to happen. If you've read Song of the Dodo by David Quammen, he writes a lot about how early settling of islands, the settlers bring in pigs and they overrun the island. They destroy the ecology of the island. Just wreck everything. Bad idea.
One of the things I want to emphasize is that, we talk about spillovers all the time, and we mostly talk about spillovers that are specifically zoonoses from animals to humans. This is a spillover as well. This virus doesn't affect humans, but it's a spillover from one species of pig to another species of pig with all of the same characteristics, where it's established an equilibrium in the native species and doesn't really bother them, and it establishes a persistent infection, and then spills over into another species and kills them.
Yes, absolutely. Spillover is not just human-centric, for sure. So there's one of the, by the way, I'm glad that Linda Dixon weighed in, because the other Dixon is not here today. She has a wonderful review article. We'll put a link to. It's in the Veterinary Journal. African swine fever, a reemerging viral disease threatening the global pig industry, and it covers all aspects of the disease. I wanted to talk just a little bit about the pathogenesis. This has been studied in, pigs in the field and in the lab. They can be paracute, acute, subacute, and chronic forms. In the paracute form, pigs die within 4 days after infection without gross lesions. Acute form can result in death of up to 100% within 4 to 21 days with characteristic pathological changes all over the place in many organs, lungs, urinary bladder, kidneys, etc. And then you can have subacute and subclinical forms in some pigs, apparently. You're infected but don't display clinical signs. And the virus can persist in them for a while, so that obviously can be a problem if you're not looking for it. The other thing I was interested in is transmission, and she writes, the dramatic spread in the Baltic states is unprecedented, and transmission mechanisms involved remain unclear. They may include movement of infected wild boar carcasses, and or environmental contamination. A role for persistently infected carrier animals has also been suggested. So,
big problem. This paracute infection is weird. It's like some sort of overwhelming shock syndrome or something like that.
Yeah,
I mean,
sounds like it. Yep. This is interesting. Shedding of virus through nasal discharge or feces seems to be limited, but virus transmission to domestic pigs and boar used as in-contact controls was effective, also inducing acute disease. Blood is highly infectious and likely to be the main source of infection for contact pigs. And then she reviews what's going on about vaccination. Development of vaccines has been deterred by large gaps in our knowledge of infection and immunity. Precise nature of the protective responses had not been determined. But she goes through all the different types of vaccines that have been tried and have not worked, including subunit vaccines, inactivated virus vaccines, attenuated vaccines, etc. So this is not without activity. So I must say that this is, I've heard of course, African swine fever and the virus. I didn't appreciate the extent of the problem. So Thank you, Tomas, for pointing it out.
Yeah.
Really great.
And Tomas, we don't really have any great advice for the big political debate over culling of the pigs.
We don't, except that there's no vaccine. That would be the best. And that's
pretty much
years.
Culling them is going to need to happen. And if if people are protesting that, well, then they need to come up with some better answer for how to deal with ASAP.
Well, clearly, it's going to be a combination of culling and biosecurity, right?
Right.
That's the way.
Yeah, the message we do get out of this is that, as Linda says, if you want to deal with this, you got to deal with the infection in the wild boar.
Yes.
Somehow.
And I will point out, by all reports, I haven't had it, but by all reports, wild boar is very tasty. Apparently, it makes good sausage and barbecue, and it's fine for all that.
So when I was in Switzerland a couple of months ago, I don't remember last year, one of the participants on TWiV was he likes to hunt boar.
Oh,
but his lab has found that a lot of the boar have trichinella infections, right?
Right.
So, in fact, I went to a group meeting and they were talking about a survey they had been doing of trichinella in various wild animals, boar and deer and so forth. So I asked him, what do? You just cook it really well? He says, no, I bring it in the lab and I test it. Make sure it doesn't have trichinella before he eats it. It can be pretty big. I saw some pictures of it, really big.
Yeah, but all the best recipes I have for pork cook the hell out of it.
Yeah, you should.
That shouldn't be a problem.
I guess he doesn't want to.
I don't know.
Back on the darting them with vaccine or something, there's problems with deer here in Ann Arbor, and so there's a deer culling thing with hunters in some of the city parks and stuff. They just finished the most recent one, but it occurs to me that starting with some kind of contraceptives is potentially also a problem in the absence of a vaccine.
Mm.
But you have to develop some kind of contraceptive, which
Yeah,
might not
and
be any easier than a vaccine.
The contraceptive that actually has been tried extensively for deer, and there's work going on because they're a huge problem. They're a native species, but they're just multiplying out of control because we wiped out all the wolves and cougars, and it's not a very good solution for reducing a population, because at best, even if you have a 100% effective contraceptive and you shoot the animal with it, that animal is still walking around doing the damage it otherwise would have been doing.
Mm-hmm.
So maybe it's not reproducing, but it's still going to be an animal in the ecosystem. Whereas if instead you load the rifle with a bullet instead of the dart, then you get rid of that animal and it's gone immediately.
Right. But we have a large population of people in Ann Arbor who are very against the shooting of the deer. So that's the thought.
That always comes up in these conversations.
Yeah.
Yeah, we do culling in New Jersey too. We have a big deer population problem. I have them running around my backyard all the time, just darting back and forth. They leap over 6-foot fences, and they multiply. It doesn't bother me too much, but they do get out of control. So you have to take care of them. Okay, we have a paper also today.
That was all a snippet.
That was a snippet.
We're now almost about to start the second hour of the show.
This is sent in by Eric, who writes, here's a really interesting paper. It demonstrates the importance of gain-of-function research to assess the zoonotic potential of viruses. And he said maybe you could get Paul Dupré on because he works on one of these viruses. But I didn't even try because he's traveling more than anyone else I know. And anyway, you need subtitles for Paul.
That's right.
No.
I do. I don't know what he's talking about. Journal of Virology, structure-guided identification of a non-human morbillivirus with zoonotic potential. And speaking of the Pirbright Institute, some of the authors are from there, University of Cambridge, University of Birmingham, University of Oxford, University of Glasgow, University of Essex, wow, and then Nebraska, Department of Agriculture in Clay Center. And we have Nursariza Abdullah is the first author. Do we have multiple first authors? I don't think so. And the last author is Delon Bailey. So this is about morbilliviruses, which is a genus within the Paramyxoviridae, including measles virus. It's kind of timely since we are having measles outbreaks in many parts of the world. You know, measles kills up to 100,000 people every year.
There's an outbreak going on right now in the Portland area, I think.
There's one here in New York City. Yes. We've had 200 cases here, mostly in unvaccinated people.
Yep.
So that is a morbillivirus, but there are lots of morbilliviruses in animals of different sorts. For example, canine distemper virus, which Paul Dupré works on, besides infecting canines, also has caused outbreaks in lions, tigers, primates.
But not bears.
Not bears?
I don't think so.
No. Then the subject of this paper, peste des petits ruminants virus, PPRV. You can tell by the name it infects small ruminants like sheep and goats. And this is a big problem for these little sheep and goats. Over 1 billion sheep and goats are at risk for infection.
I'd like to jump in and give the overall importance here of this paper and say that we've had lots of discussion on TWiV before about what is involved in viruses jumping species and doing experiments where you have viruses gain function that allows them to now infect a different species. And these people are looking at that question with respect to morbilliviruses, and they're doing it in a way that they're not having to create the viruses themselves.
Right.
And so that's the importance of this paper that's in Journal of Virology.
Within the morbilliviruses, that's a particularly serious concern because measles is one of the most contagious viruses known, right? Mm-hmm. So if you made a canine distemper virus or a PPRV, petit ruminant virus, that could infect human cells or humans, and that you could really cause some serious problems.
And measles itself, right, is thought to have been a spillover from rinderpest, right?
Mm-hmm.
Right. And one theory I saw was that this might actually date to the domestication of cattle.
Yep. So the point here, there are lots of morbilliviruses of different animals, including whales and seals and dolphins. And the point is that they're all genetically very similar, and they all bind the same receptors, of course, in their own species, their host species, but they bind the same receptors, and there are two different ones called SLAM-F1 and Nectin-4. And so whether the morbilli is measles or PPRV or dolphin one, they bind these receptors. And so that has led to spillovers. And for example, canine rinderpest is a virus, that rinderpest virus, a morbillivirus of cattle, recently eradicated. So now we no longer immunize cows against it. There's worries that some of these other viruses will spill over, canine distemper and PPRV, because they're so similar. Right. And previously, there's been enough cross-reactivity between the antibodies produced by rinderpest vaccination that prevented that from happening. But there's worry that that's not going to happen anymore. So that's why there's worry about zoonotic spillover. And as Kathy said, one way you can look at that is take animal isolates and passage them. If you want to know if a PPRV can infect human cells, you could passage it in human cells. And that's been done. And you can select for gain-of-function mutants that now can infect human cells. And probably that wouldn't be permitted in certain places. Here in the U.S., the restriction has been on, influenza and coronaviruses. I'm not sure if CDV is or PPRV is in it, but you can imagine that people would get upset about it. And that's why in this paper they try to use experiments that don't involve infectious viruses. So you don't actually generate a virus, a PPRV that can infect human cells. They do other things, but in the end, I think you have to to really get the answer, right. to kind of presage what's going to happen at the end there. So a series of very interesting assays, which ask the question: could PPRV infect human cells? Does it have the potential at different steps, at virus entry into cells and RNA replication? They look at those two steps essentially and ask whether it can. And so the first experiment, they take a cool assay. So these viruses infect cells by binding to the receptors, which I noticed before. SLAM-F1 is the one they look at here. The virus has two glycoproteins, H and F, hemagglutinin and fusion protein. They produce both of those proteins on one cell, and then they have a second cell that produces the receptor, and both cells have different fluorescent markers in them. And you mix the cells together, and if the HF proteins can bind the receptor and induce fusion, the fluorescent proteins will mix, and you can measure that.
Yeah, it's actually that they have a split in the protein that in a split in the luciferase protein and a split in the GFP protein, and they don't do anything with the GFP protein coming back together, but they do assay the luciferase coming back together. So they have half of the protein is actually the way I understand it, both halves of the protein are in both of the cells that are going to fuse, but it's only the fusion that then allows the GFP to form and fuse and then bring these two GFP to sort of reassemble, and then the two parts of the luciferase come back together too. It's not very well described even in the paper that they reference.
Exactly.
But I was disappointed that I didn't think they described the assay very well in materials and methods, and I thought they should have described it in the main part of the paper because it's one of the main assays that they use throughout the entire paper.
Kind of the core of the paper, yeah.
I agree. I mean, essentially, it's to measure the fusion of the two cells. Right. It's a really neat assay, then
it
they
is.
should really describe it. So if you
Now,
take,
should we go through what these two different receptors do in the host for the virus?
if you'd like, it's not essential, but you could.
No,
You
just
could.
real briefly, because I think this is really cool. When Paul told me this story, I was like, wow, that's neat. The SLAM receptor is often referred to as the entry receptor for the Morbillivirus. They're both entry receptors. They both let them into the cell. But in terms of the host, if you inhale measles virus, it binds to immune cells in your lungs through your SLAM receptor and then forms the systemic infection. Once the systemic infection gets going, you've got measles in your bloodstream, and it can then bind the Nectin-4 receptor, which is on the inside of the cells, the epithelial cells that line your lungs. So that gets it into those cells, and when it comes out of those cells, you exhale it.
Right.
So it's got a mechanism that sends it into your bloodstream before it can get back out through your lungs. So Nectin-4 is sometimes called the exit receptor, because that's going to get the virus to its next host.
Yep. Now, in this paper, they just focus on SLAM.
Just looking at SLAM.
But it's a cool story. So if you take the HF proteins of, say, PPRV, put them on one cell, and then the other cell you have a sheep SLAM, those two cells will bind and you will get fusion. If you take a measles HF and you have human SLAM on the other cell, they'll bind and fuse. You'll get fusion. But if you have a PPRV HV and, say, a human SLAM on the other cell, they won't fuse. So there's a barrier to PPRV HF mediating entry into the human cells. So that's the basis of the assay. And what they do is they look at the sequences of the H, the hemagglutinin protein, and they can see areas of conservation and areas that vary highly, and they narrow down to a 4 amino acid stretch, PTTIR, and they take that sequence, that 4 amino acid sequence from measles and substitute it into the area of PPRV, and that allows fusion to occur. And in fact, there's just one amino acid there that is enough to get PPRV to bind and fuse with cells producing a human SLAM, and that's 191, the amino acid at 191. Single amino acid change. And none of these changes affect the ability of the PPRVH to bind the sheep SLAM. And they don't affect protein stability. All of that means to them that they're maybe neutral. So these changes could happen in the wild, allowing PPRVH, say, to bind to SLAM on human cells, and they would remain evolutionarily competitive because of that. Whether that would
That's
happen.
pretty scary.
It could be. Be scary.
Single amino acid change.
Single amino acid change.
Yeah.
Yes. And they confirm that in different models of entry. They have a pseudotype assay where they take HIV-1 particles, they put the PPRV-F and H glycoproteins on the surface of those HIV, and these are HIV that don't replicate. They can then ask, will this infect cells that have human SLAM on them? And the answer is yes. So all that tells you that at the entry step anyway, 1 amino acid is all it takes to get PPRVH to bind human SLAM and get into cells. And of course, lots of other things happen between that and the end of the reproduction cycle, right? So what they look at is RNA replication. They have a mini genome assay where they cut out most of the structural proteins in the RNA genome, and they just have the proteins needed for replication on a genome, and they have a reporter molecule on that genome. You put that into cells, it will replicate, and you can measure the replication by your reporter. And they say that if you take this mini genome from PPRV and put it in human cells, it will replicate. So at least the RNA replication machinery is of PPRV. Without any modification, it will work in human cells. Now, there's still more steps, of course, between PPRV, even with a one amino acid change of getting into human cells and replicating and making infectious viruses. And then in a host, a human host, there's even more. There's an immune response, there's spread within the host and so forth. So this doesn't mean that automatically one amino acid would be enough to make PPRV a human pathogen.
No.
But it does raise some concern, right? You should probably be looking at what's circulating out there. And that just reminds me of the H5N1 story where Fouché and Kawaoka separately adapted H5N1 to be aerosol transmitted in ferrets, a real gain of function, right? They made the virus. They were crucified for it. And they said, well, we could look for these amino acids changes in nature, and they got yelled at for suggesting that. Why would we want to do that? We don't need these experiments. Well, here it's the same idea, and it's worth looking at these changes in nature to see at 191, are we getting anything that could allow infection of human cells? I think that's really worth looking at. They went on to do a couple of interesting things. They took this position, 191, and they randomly mutagenized it to see what would happen to infectivity, to the fusion assay, I should say. And they show that other amino acid changes at this position can overcome the restriction to fusion to human SLAM. They mutagenized the receptor as well, and they're trying to understand how these changes in H overcome binding restriction, and they can, by mutagenizing the receptor, they can get an idea of what's contacting one another. The part that I really thought was cool, they looked in the human sequence database.
This was neat.
To look for polymorphisms in the SLAM, the gene encoding the SLAM receptor, right, for these viruses. And of course, human SLAM, you look in the human database, they can find lots of changes in this database. And they identify a number of single amino acid changes in SLAM that reduce fusion mediated by measles virus H and F. And so, in other words, there are people out there that may be resistant to measles or somewhat resistant to measles due to changes in the receptor. And I think this is something that we've kind of touched on right here on TWiV on and off, this idea that mutations in specific genes can influence susceptibility to infection. This one is interesting because it's in a receptor for the virus.
Yeah.
So not everyone is going to be equally susceptible, and one reason may be changes in the receptor itself. The other interesting aspect, which, and this is the last experiment, mentioned earlier that there's cross-reactivities among or between these different morbilliviruses. And they do an experiment with collections of human serum, and they neutralize, they attempt to neutralize viruses of different sorts, measles and their PPRV, and the PPRV with the 191 amino acid change. So human serum will neutralize to a certain extent PPRV. They don't actually use PPRV, but they use an assay that would measure it. And the 191 amino acid change in the H glycoprotein reduces the ability of the sera to neutralize that virus. Certain human sera failed to equivalently cross-neutralize the R191 virus. So in other words, if, PPRV out there at some point changed that 191 so that it could infect human cells, not only could that be an issue, but at the same time, any protection you might have, say, because of measles, previous measles infection, or maybe measles vaccination, would also be overcome by that amino acid change. So it might be. It's in an important antigenic site. So that's two concerns for that, right?
Yes.
Overcoming tropism and overcoming antibody neutralization. So that's all done, again, without making virus that would raise any gain of function concern. It's using two kinds of assay, one for fusion and one for RNA replication, to suggest that this PPRV could at some point infect human cells, and who knows what could happen after that.
Now, there is a little bit of, as you pointed out, being able to get into cells, being able to replicate in the cells, those are only a couple of pieces of a complete infection.
Yeah.
And I think it's worth pointing out that there are some data that suggest it's not going to be quite that simple for the virus. My understanding is that transgenic mice that express the SLAM and or Nectin-4 they don't really develop measles, right?
That's right.
So they've got the receptor, the virus can replicate in their cells, but it doesn't get the job done. So there's more that probably has to go on. Also, a single amino acid change, I suspect this has happened in nature.
Right.
You've got gazillions of these viruses out there, and they're not replicating perfectly. And this is a single amino acid change. It's probably happened before. And you've got these animals that are in close association with humans. So if this was a bomb waiting to go off, it already would have. But
that's my thinking. Exactly.
But at the same time, if you want to monitor this virus in the wild, this would be a good mutation to look for. And if you see it popping up and you see it persisting, then maybe investigate a little further in that.
So that's exactly what Peter Palese says about avian influenza H5N1. He said, it's been infecting people for 50 years. If it were going to explode, it would have. But there can always be a time, right? It could happen. So
true.
I'm not sure that's so comforting to people, but I do think that we have a clue of what to look at here, and we could say, hey, if something arises, all of a sudden you get at 191 this exact mutation, we should watch out for it, right?
Right.
I would say that if there's going to be a problem, this is probably going to be part of the problem.
Right.
Yes.
To be part of it, right?
Yes. This is likely. This could be necessary, but it probably isn't sufficient.
Yeah.
Right.
They actually say there is no clear indication that enhanced entry alone is sufficient to confer a pathogenic phenotype to PPRV in humans.
Right.
Mm-hmm.
And in fact, they cite an experiment done in ferrets. They made canine distemper, which could not bind to SLAM. They gave it to ferrets. It reverted in the ferret to be able to bind SLAM, and it still didn't restore virulence.
Right.
So, yeah, there are other factors.
They are not trying to pull the wool over our eyes.
Not at all. No, I don't think they're trying to scare us either.
No.
But they say at the end, we encourage epidemiological surveillance of mutations in this region, akin to the surveillance for influenza virus.
And they've come up with a cool approach to look at these types of things without having to create viruses that set people off.
Yes.
For want of a better way of phrasing it.
Indeed.
But at some point, we are going to need to go to a model that really does the gain of function and look at transmission to human cells or primates or something like that if you really want to probe this problem.
Yeah, I think it would be interesting. I mean, now I'd like to know if PPRV with this one amino acid change, if it could go through the whole cycle in human cells in culture, right?
Right.
But I guess they don't want to do that.
Right. I would think that experiment should be done in some kind of containment. I don't know if it requires BSL-4, but maybe 3.
Yeah. That's what the ferret H5N1 experiments were done under.
Right.
I think that's probably sufficient. I like the way they put this. We wanted to see if we could predict gain of function mutants a posteriori.
Yes.
It's great. A posteriori, without in situ derivation.
So that's where they pulled the data out of. No.
Anyway.
Well, I'm trying to figure out whether Pirbright has BSL-4 or only BSL-3, but I haven't found it quickly.
They must have BSL-4.
I think they have 4. This is where do you need 4 for foot and mouth? Does anyone know? Because I know they used to work on it. And they had a leak once. Some pipe was leaking and 4 got out, and that was a big problem years ago. We did cover it, yeah. So I think it is a 4. So they could do it there if they wanted to.
And actually, in the UK, they call them containment levels. So it's
Yes.
CL3 and CL4.
Uh-huh.
Right.
So it's interesting, and I thought, in light of our interest in gain of function, right?
Yes.
We have some interest, not in doing it, but actually talking about it. Okay, thank you, Eric, for that. And I don't know if we've done a measles paper before. We certainly haven't done PPRF, right?
Didn't we cover that the receptor thing that
Maybe the Nectin story.
Roberto
Cattaneo, yeah.
Cattaneo
Maybe. Okay.
Yep, I found it. Pirbright seems to have 4.
They have a 4?
Mm-hmm.
Okay. I like your notes here, Kathy. It's like stream of
Yes.
conscious.
Yeah.
I don't get this. Oh, there it is. Oh, yes. Okay.
Yeah.
I had some issues with it. There are some sentences in this paper I absolutely could not understand, reading multiple times. But
Yeah, there's one in the second figure legend that's okay. It's in the 4th line down of the figure legend. Of note, a number of specific amino acid residues were found in more than one site, Y524 and Y552. And then you look at that part of the figure, and I can't figure out what they're trying to say. So, yeah, that was one of my hang-up sentences. So, oh well, we got the big picture.
Yep, that's the big picture. Thank you, Eric. And this was a listener-driven TWiV today, both the snippet and the paper. And the paper was shorter than the snippet.
Yes.
So maybe we should reverse the labels. All right, let's do a couple of emails. Rich, can you take that first one?
Anthony writes, Professor Condit noted that the current Ebola outbreak was not getting much play in the news. As long as Ebola was what Dr. Hotez called, quote, an imaginary disease of white people, end quote, and worried the comfortable here, media marched. As it became increasingly obvious, despite the best efforts of Dr. Osterholm, that Ebola afflicted the poor in Africa and was not going anywhere else, the American public no longer cared, for what it's worth.
That's pretty much it.
Same. I think the same has happened with Zika virus, right?
Yes.
Yeah.
Congress, if you remember, Congress appropriated some money a while ago before it became clear it wasn't really going to go anywhere in the U.S. It's unfortunate. And Scalca writes, what a touching and uplifting discussion with Roland. I know he has been much admired by his colleagues, and now I know why. Not only an outstanding and accomplished scientist, but a wonderful human being. Thanks, Anne.
Yeah, that was a really nice interview. I enjoyed that.
Yeah, it was fun. And Roland wrote to me, he said, you should come visit me in the woods. I haven't finished talking. I want to talk to you some more. It was fun. But it's 4 hours north of Madison.
Wow.
That is in the woods.
By the way, Kat, when I went, I flew to Milwaukee and I drove to Madison. That was really pretty good, actually. Although I drove at night on the way, and that wasn't so good. But coming back in the daytime, it's not a bad way to get to Madison. Mm-hmm. And if I visited Roland, I would fly to Milwaukee and drive 5 hours north or whatever it is. Kathy, can you take the next one, please?
Sure. This is from Anne with an E, so a different Anne. Professor Racaniello and TWiV team. The most recent episode of Inquiring Minds, a science podcast I listened to, contains in its most current episode, January 8th, a discussion between the hosts about a topic I've heard discussed on TWiV, pricing and access to scientific journals. The title includes a mention of the New Horizons probe. It's the last segment of the episode, starting at about 10 minutes, 45 seconds from the end, and it references the University of California system versus Elsevier. Many points are similar to points found in your previous discussions of this, but there's one that's different. I don't know if you've covered the story already. Feel free to ignore it if so. And Anne gives the link to that. And then also a link to an article about the dispute. There are many others out there. Regards, Anne. P.S. The podcast is also available on the podcast app. So I didn't check out the podcast yet.
I listened to it, and it's the it's most of the points that we've already discussed. I'm not quite sure what she's talking about the point that's different, except that one of the hosts makes a big issue of saying that the scientists are guilty in part of driving this by perpetuating this idea that your promotion is contingent on publication in high-profile journals, and so they're driving the consumption of journals that people are charging a lot of money for.
I think that's right.
Yeah.
Yeah.
And he takes issue with the notion that people say, well, there's nothing we can do about it, because that's the way it is, or something like that. And I agree with him. Of course, there's something you can do about it. You just have to change the behavior. And every individual can behave in the way that they think is right and make their points and behave appropriately. And slowly the ship will turn.
So apparently, Alan, the UC system is trying to fight Elsevier.
Yeah.
Yep. They don't want the whole UC system. We're talking about $10 million worth of subscriptions.
Yep.
And what do they want to reduce it or something?
They want to. Yeah. They want to change the whole subscription fee, open access fee structure.
Yeah. Yeah. Well, there are other state systems that could do that as well. Not as big, I guess, but if they all joined in, that might help, right?
Yeah.
Then so same Anne wrote in, no doubt your team has heard about this apropos of Professor Despommiers' comment on a recent podcast episode. The Cold Spring Harbor severs its ties with Watson.
Mm-hmm.
And I was talking to someone about this, I think it was at our last book meeting, and they said, well, he made these comments 30 years ago. Why didn't they sever their ties with him then? And that's a good point, right?
Yeah.
Because he was bringing in grant money and fame, and
yes,
they figured they could just ignore it.
Yeah,
he didn't just suddenly get like this.
No, he didn't.
When I read this, Mike Wigler's comments at the end said Dr. Watson's views on race were not newsworthy in the first place. It's not news when a 90-year-old man who has lost cognitive inhibition and has drifted that way for decades as he aged speaks from his present mind. It's not a moment for reflection. It is merely a peek into a corner of this nation's subconscious and a strong whiff of its not well-shrouded past secrets. So it alludes again to the sort of aging issue, but as you just said, Vincent, this is things that he's said many years ago as well.
Yeah, I am not. I'm done with this excuse making of, he's old, he's losing his mind. No.
I don't necessarily think it's making an excuse.
I didn't think
it's making a comment that said it's even more pronounced. There's less.
He's become less inhibited, but he was never not like this.
Right.
I guess who aired that? PBS, the interview, right?
Yes.
They were just trying. They knew it would be controversial, right? And that's why they did it. And we know that's what happens. But all right, it's over. The next one is for you, Kathy. Ken.
Ken writes, "Hi, TWiVers. Although my career has nothing to do with biology, I've been listening to TWiP and TWiP for several years now, along with other science and skepticism podcasts, out of pure scientific curiosity while I spend my working day doing CAD, GIS drafting, and design work, and 3D modeling work for a geotechnical engineering firm in Vancouver, BC, Canada. I can't say that I have that great of an understanding of your topics, but it's fascinating to see the scientific method in progress. You've improved my vocabulary sufficiently so that when my daughter was born at just 29 weeks and 1.5 pounds, and with a number of complications, including a rare genetic metabolic disorder called MCADD, I think I had a much easier time than I otherwise would have understanding what the doctors, nurses, and specialists were talking about over her 5-month stay in the NICU. She's 6 now and doing great. MCADD is manageable by diet, and she's had no developmental setbacks. Her older brother is a fan of the show The Monsters Inside Me. Anywho, in TWiV 530, there's a discussion about words that are portmanteaus. I believe I have the ultimate example with a built-in pun. Perhaps it is a meta portmanteau. It's hilarious to me, but it seems to get no better than a groan from everyone else. Here in Vancouver, we have a large bridge over the Fraser River named the Port Mann, and man is spelled M-A-N-N. A few years ago, it was replaced. Once the new bridge was built, the old bridge was taken apart, lowered down piece by piece onto barges to be hauled away, in a process that I've named the portmanteau.
Nice.
Thanks
Very
for
good.
all your great science.
That's great.
Yeah, I liked it. Definitely.
Speaking of taking apart bridges, the Tappan Zee Bridge was just replaced. It's a big bridge that crosses the Hudson River about 30 miles north of the George Washington Bridge. And they blew up the old one. So the old one
literally blew it up.
Well, yeah, the old one was this metal span thing resting on concrete pilings, right? And they put charges on the 4 pilings and they detonated them, and the whole thing just dropped into the river.
Wow.
And you can find a video of that on YouTube. Should have made it my pick, actually. And I was like, why wouldn't they just take it apart? Because now they're going to have to go in the water and pull it out, I guess. But this one makes sense. The old bridge was taken apart, lowered down piece by piece onto barges. I would think they would do that. But
Are they going to take the old Tappan Zee out?
Maybe they're going to leave it in the water, but isn't it a problem for boats, right?
I'm sure they did that calculation, and if the shipping channel is not going to be restricted as a result, then
maybe leave it there.
Leave it.
Wow.
Artificial reef in the river.
Okay. There is a short note here from Anthony. Really interesting. It's a NewJersey.gov article. Cape May County horse found to have rabies. Now, this is nothing new. This happens. But the article is interesting because it gives you a lot of information about rabies. So this is a Cape May County horse was positive for rabies, was on a farm in a rural part of the country, was having neurological symptoms. It was tested, had rabies, 20-month-old colt, and it was previously vaccinated.
Yeah.
Interesting.
Yeah, that's the weird part.
So they revaccinate everyone. That's interesting. And then,
how effective is rabies vaccine in horses? Is this a known?
I don't know.
problem?
Don't know. They don't mention that. But they do say rabies is endemic in New Jersey and often detected in bats, raccoons, foxes, skunks, cats, and to a lesser extent, domestic livestock. Transmission is almost always from the bite of a rabid animal. Virus is shed in saliva several days before the onset of clinical signs. And so presumably one of these animals contacted the horse in some way. And there's a number here if you want to report suspicious rabies in species other than livestock. So thank you. That's cool.
Yeah.
Right here in New Jersey. All right.
Rich, well, interesting anyway. I don't know about cool.
Yeah, it's not cool. You're right. But I use cool to mean interesting.
I do too. I have to catch myself on that sometimes.
And students say it's insensitive at sometimes. But yes, I understand. It's not sensitive, right? All right, let's take the last one. This is, I think, Rich. Can you take that one?
Sure.
And there's some curse words. Be careful.
I think I got them. Anonymous writes, "Hey, I was listening to your December 13th episode about Hanta, and it just so happens that an outbreak was happening and is happening in Argentina in a town called Epuen. Contrary to what you said in the podcast, this strain of Hanta can jump between humans, apparently. From what we know, it started in late November when a farm worker got the virus from cleaning an old shed, literally a textbook case in my arrogant opinion, IMAO, then went to a birthday party and a bunch of attendees got it from him, including a 14-year-old birthday girl who is now dead, R.I.P. The farm worker is fine, though, and you can imagine being him now. Poor guy. This human-to-human transmission isn't new, though, and at least according to Spanish Wikipedia, there was an outbreak in 1996 in El Bolson, really close to Epuen, 25 miles, that also affected some medical staff treating the patients. Some died. So, yeah, some evolution trickery going on down south. The latest tally I can find says at least 26 cases, 10 dead, including a nurse. coincidentally, you guys mentioned on the podcast that the place-based naming, you mentioned the place-based naming and how inconvenient it can be. Epuan is a beautiful mountain town that lives off of tourism, and January is high season. It's messing up their economy, sadly. So there you go. Yes.
So it turns out there were 2 outbreaks, as he said, and I found articles about them, and they both involve person-to-person transmission. Apparently, there's even a CDC article, EID, Emerging Infectious Diseases, on the last one, 1995. So I still would say that person-to-person is unusual for Hanta, but not unheard of.
Not unheard of.
Right.
We've now heard of it.
We have now heard of it. Thank you.
And if you look up Google Images on Epuan, which I'm probably not pronouncing correctly, it is indeed a beautiful place.
And I suspect is maybe the pronunciation, but yeah, it looks gorgeous.
Okay, time for some picks. Alan, what do you have this week?
I have a database, which doesn't sound that exciting on the face of it, but this is the Retraction Watch database. And I've linked to the blog article where they launched it. This came out a few months ago. It's a database of over 18,000 retractions of scientific papers. And they talk in this blog post about what went into building this and point out you can't just pull retractions out of PubMed. You can't just do search all the retractions. It's publications do a very poor job indexing these, either deliberately or accidentally. And as a result, this was a huge load of work, and they now have this database, which they are continuing to update. And they also link to an article at the end of this blog post. They link to an article in Science, a long package of stories and graphics of data mined from the database that shows some interesting trends and phenomena out of it. So if you are interested in this phenomenon and it's worth keeping track of, you can go and read the instructions for this thing and search. And they are hoping at some point, if they get the funding to do this, to create a program interface, an API, so that people would be able to mine the data more directly.
I'm so sorry this even has to exist.
Well, of course, but science is done by people.
I understand.
And, you know, if we're gonna have people doing something, there will be some who will screw it up, and it's good to be as open as possible about that and have it as easy to study as possible.
Absolutely. But still,
yes,
you would hope people could be better. But what was I thinking?
Yeah, they're people.
Well, you know, Arturo Casteval, and we've talked about his articles on this. Covered it a lot. You know, there are mistakes, and then there's real fraud where you got to do something to get your next grant, and so you make it up. It's really bad.
And a critical part of this Retraction Watch database, it's not just that they collected all the citations to retractions. They actually went through and hand-coded the reason for the retraction when it was discernible. So it can actually be separated into stuff that was retracted because, you know, hey, turned out the experiment didn't work that way.
Yeah.
You know, and that does happen. And you can distinguish those from the frauds, and the science articles actually have a good set of graphs showing this.
There's one paper which I think needs to be retracted by the principal investigator author and has not been. And that's, of course, Andrew Wakefield's paper.
Yes,
it's been
and
retracted
it never will
by the journal. He has not retracted it. He never will. He should, and I think that would really help with this anti-vaccine. You don't think so?
It's never going to happen.
He's still making money off of it.
He has his audience. He's got his followers. And even if he did, I think at this point it's bigger than him.
It's very sad.
I think people would make up conspiracy theories about how the government or the pharma industry got to him or something, and they would continue to soldier on against vaccines. It's
You're probably
become
right.
its own thing.
You're probably right. Okay. Rich, what do you have for us?
I'm looking through Retraction Watch. So I got to pull back. Yes. A couple of months ago, I think, on public television, was aired a new Ken Burns documentary called The Mayo Clinic, Faith, Hope, Science. And it was absolutely outstanding. It's the history of the Mayo Clinic in Rochester, Minnesota, and now elsewhere. And it's very well done, fascinating. I have some affinity for this because my wife's father was there for 13 years. My wife grew up in Minnesota because of that, in Rochester, Minnesota. But, it talks about, a country doctor and ultimately his two sons who, seeing a need, ultimately built up this clinic. And it describes the uniqueness of how it functions. All the doctors, all of the staff, doctors and everybody are salaried, okay? So your compensation has nothing to do with how many patients you turn over. All of the disciplines are kept in close contact with each other. So every case that comes in is approached from quite a holistic perspective. For example, one really interesting thing is that I don't know how often this is done, but it was, I guess, initiated at the Mayo Clinic and still goes on there and probably goes on other places as well, is that they have a pathology laboratory directly adjacent to the operating room, so that for any resection that's being done, like for a cancer, they can have a pathologist look at it in real time and render an opinion as to how serious it is, whether they've got the whole thing, and so that the surgeons can deal with it right while the patient is still there on the table, rather than having to go back and do something a week after the pathology report comes back. So I recommend this. It's a really great documentary.
And, of course, the Mayo Clinic also has a research wing, right?
Mm-hmm.
And Roberto Cattaneo and others are part of that. And he, of course, we talked about the Nectin work that he's done, and they are hiring at the moment. Kathy, what do you have?
I picked a photography contest from the University of Michigan EEB department, and I liked it, first of all, because the winning photo is just a great picture of a frog. And the pictures have explanations that are much more lengthy than you usually see in a photo contest. And so there's second place is a snake, third place is something in the UP, Upper Peninsula of Michigan. And I just thought it was a small number of pictures by pretty much amateur photographers that are really quite good. And
this
unfortunately,
go ahead.
Yeah, the frog is really great. He's my favorite, and I guess a frog won the contest last year too. But it's too bad because Dixon will never see these. But I thought he would have enjoyed them too.
They have a vervet, which Brianne sent us a picture of vervets from South Africa. She said they're not grivets, but they're vervets.
Yeah, this one is particularly artsy.
It's cool.
Yeah,
I like the snake too. It's pretty neat.
Mm-hmm.
Yeah.
This frog is really Kermit.
Yeah.
It's cool.
So,
nice.
Yeah.
I have a short piece in the New England Journal. Journal of Experimental Medicine. Sorry. I don't know if it's open access. Can you check it out, Alan?
Let's see. JXMed generally is.
I can get the whole thing. I'm not.
I think all their stuff is open access.
Wonderful.
Yeah.
It's a viewpoint. Vaccines, an achievement of civilization, a human right, or health insurance for the future, by Rappuoli, Santoni, and Mantovani. Bet you can't guess where they're from. Vaccines? Yes. I once had a photo taken, and Reno Rappuoli was part of it. And except for him, I would have been the shortest person in the photo. Vaccines have made a key cost-effective contribution to the prolongation of life expectancy and quality. So this article basically goes over why vaccines are important and why we have to defend them. talks a little bit about this book by Philip Roth, Nemesis. Has anyone read that?
No.
Mm-mm.
It's an account of the drama of polio in Western countries. I didn't know that. And he says vaccines are great, but they've been so great that nobody worries about infectious diseases very much anymore. And you take it kind of for granted that, they're not longer a problem, but they are. And he calls it the vaccine paradox. And he said, why do people object against vaccines? And he said, one of them is that they're victims of their own success. The other, fake news about the connection between vaccination and autism fostered by social media. They enjoy credibility from lay people to parliament and government levels. A widespread belief that natural is good has led to the misconception that diseases such as measles are a sort of workout for the immune system, while the fact that pathogens are professionals at suppressing immunity is widely unappreciated. And here's a good one. Finally, there's no perception of the fundamental concept that although we are immunologically unique as individuals, we are a community in terms of resistance to infectious agents. We argue it is our social responsibility as a scientific community to stand and promote understanding of the value of vaccination. So that's what we do here on TWiV, and given the current measles outbreak, I thought that would be a good pick. And you can never promote vaccination too much.
Right.
We have a listener pick from David Spector, who's our friend, Professor Emeritus over at Penn State Hershey. I meant to send this link last week when it was published, but life got in the way temporarily. Yes, it does tend to do that. It's not about virology per se or even biology, but it's a wonderful defense of curiosity and the pursuit of useless knowledge that is at the core of the love of science for many of us.
He puts useless knowledge in quotes.
Yes.
I don't think he thinks of it as useless. He's just quoting. Yeah.
So there's an opinion column by Brett Stevens, and it's about an essay written in 1939 by Abraham Flexer called The Usefulness of Useless Knowledge. And it's all about exploration in science. And he says sometimes, you end up with nothing, but often the curiosity is what pushes it all forward. And in fact, that essay was my pick, I think, on TWiV a while ago, last year sometimes.
Yeah,
so
this is familiar.
It's you can reach this. This is accessible online, the original 1939 editorial, and it's really good. It just promotes scientists being curious. And he says, we're going to waste some money, but in the end, it's okay. So this is an editorial which basically promotes that essay. So it's cool. It's important for people to know this, that it's important for us to just be curious, not necessarily have to figure out something, right? Science works both ways very well. TWIP. No, this is not TWIP. This is TWiV 532. If you listen on your phone or tablet on a podcast player, please subscribe. And that way we know how many people are listening. And if you like what we do, consider supporting us, microbe.tv/contribute, and send your questions and comments to TWiV at microbe.tv. Kathy Spindler's at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun. And I was just reminded that I needed to like the last couple of episodes on my phone. I'm doing that now. Yes.
Wonderful. Thank you. Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.